Okay. Our presentation tonight is ID tips for the blues. This will be a presentation by David Droppers, and many of you know him, of course. He has been teaching courses on topics from birds to butterflies to trees to microorganisms for a variety of groups, including college biology courses and nonprofit environmental groups. He has put his expertise to work for the many educational and public groups, and many of you probably recall the presentation that he and Phyllis Reed gave us in December about how David has helped the U.S. Forest Service with his work on the Mountain Loop Highway in the North Cascades. We are so happy to have David back to share his effervescent enthusiasm for the natural world, and especially for some of the tiniest butterflies. So thank you so much for being with us tonight, David.
Effervescent, huh?
I think so.
That's a good one. Effervescent enthusiasm. I might steal that for future descriptions. Now I'm here primarily because Pelham was supposed to give the presentation, but his computer went kaput.
But we've been talking about you doing this for a while. It's just that you had given a recent presentation, but so this is
yeah. I mean, I
been in the works.
feel a little bad. Like 2 of the last 3 are me, but as I promised you, if anything like that happened, a speaker had to cancel. Hey, we've got a lot of beginners, intermediates, and people who just like refreshers prior to the season. I mean, it's March 1st. People are going to be seeing the first butterflies this month, so you know. Time to polish up the ID skills. All right, let's see here. Work on sharing. All right. Once again, Paulette, you can see my cursor, correct?
I can see your cursor.
All right, and I need to move. And move all this out of the way for everyone. And I thought there was a way to do that. I think if I just leave it alone, it'll go away. Okay. So yeah, so I'm actually getting a little nervous now because this is supposed to be butterflies of the blues of Washington. And I see Sandra here from Oregon. I see Christy from Montana, Jeff from Idaho, Glenn Fine down in California. So it's like, oof, this is not going to be a complete list for you folks. You got blues that we don't got. So hopefully this, as Paulette says, gives you just, this is kind of where you look for different blues, or this just reminds you of at least some of the blues in your area. But yeah, we got a lot of blues to cover. So many blues. Oh, hold on. Gotta click back on the screen. There we go. So just to back up, big picture here. The blues are are a member of the Lycaenidae, also known as the gossamer. So this includes our coppers, our blues, our hair streaks, and also our harvesters, but we don't have any harvesters over in the West. That's a east side only butterfly. There's about 5,000 species worldwide, give or take, with only about 150 in North America. The larvae are considered kind of slug-like. They don't really have the extended prolegs or really identifiable heads. Only 4 instars rather than the usual 5, or you've got 6 in the greater fritillaries in the Spirea or Guinness, whichever one you want to use there. Many of these species are attended by ants in the larva stage. So it's called myrmecophily, where the ants kind of act as protectors in exchange for basically a honeydew-like substance, very similar to what they do with aphids. So big picture, blues are a member of this group. In general, blues kind of buck the trend with most butterflies. So most butterfly groups are more diverse down in the tropics. Blues are actually more diverse up in temperate regions than they are down in the tropics. We have 17 species in Washington. The taxonomic opinions will differ on this. That's why I say it's kind of fortuitous that we're doing this online. If we were to do this in person, there would be black eyes as soon as we start getting into the taxonomic differing opinions there. And yeah, most of these butterflies are myrmecophilous. So once again, the larvae attended by ants during the larva stage, and different species to different degrees, I would say, is a pretty good summary. Males by and large are blue, females by and large are brown, although there's a lot of variation between the females. So I get a lot of people kind of going, oh man, the blues, the males are more showy than the females. Remember, the females want to live a little longer, lace more eggs, so it behooves them to not stand out as much as the males. The males still got to attract the ladies, so they still got to be pretty showy, but really strong sexual dimorphism in the blues. So topside might be pretty, but on the top side, there's a lot of kind of the same thing going on here, although you'll see plenty of differentiation. It's the ventral side, the underside, we really want to look at to identify these blues. Although topside can help, a lot of the really good information is on the bottom side, and I think kind of the more interesting side anyway. So just to tease you, I'll say, here's a video from one of my biology classes with as far as how many blue butterflies there are. This will kind of get us excited for the upcoming field season. Everyone sees them all, right? Let's watch that again, just because, like I said, it's been a long winter and I can't wait to see this again. I mean, hundreds of males there, all puddling.
It just kind of looks like snow there for a
Yeah,
second.
it does, but nope, those are all blues. So here's your first blue. This is a blue that confounds a lot of people because it doesn't quite have any blue features, but it certainly is a blue butterfly, correct? Just for fun, I'm going to keep the chat open. If you want to test yourself, I'll have a little sip of water before everyone. If you want to throw your guess as to which species that is in the chat, we'll see, and you can test yourself, see how many of these you get right. Let's see. Ah, you guys are correct. Okay, cool. It's not a blue. Yep, another correct one. Yep, not a blue. It is actually our blue copper. Let's see. Wish there was a way to hide that bar, because I think you guys, since I'm sharing my screen, you can probably see that bar. Anyway, blue copper. So the male, brilliant blue above. I don't want to play favorites, but this is a great butterfly. I mean, those males are stunning. Those veins, though, the dorsal vein, well, I shouldn't say dorsal veins on the underside too, but dorsally, the veins are lined with black scales. That's not going to be the case in the other. So if we back up a bit, you see on that male blue, you can see the veins, but they're all overlain with blue scaling. Whereas, oh, what the hell, one more time. Oh, I can't wait. Whereas, yeah, on the blue copper, they are lined in black. You can clearly see them. These are often a lot larger too than your average blue. You're gonna think, wow, that's a big blue fly in there. That's your first clue. It's a blue copper. The females are brown with black spotting. Really can't confuse those with any other blues. There's no female blues with that much black spotting on there. Maybe the occasional discal cell spot, but that's about it. Below is usually, I say usually, lightly too unmarked. There's a couple of populations, I think down in Oregon, that are pretty strongly marked actually. But you might see this just completely whitewashed underside of a blue copper. They do have a single generation in summer. Their hosts are buckwheats, which once again kind of lends them to be confused with blues, since they're around one of the major host plants of a lot of blues. So they commingle with blue butterflies. You generally find them in more arid landscapes from low to high elevations. I've seen them high in the Cascades. I've also seen them down in the Sage Steppe. But brilliant butterfly, not a blue, but one to keep in mind because it can throw you for a little bit of a loop. Now, here's another one you can take a guess at. Now, these are in no particular order. and largely because you're not going to find these blues in any particular order. Now, that's just a bad excuse for me not deciding what order to put these in. That's really the honest answer there. Anyone got a guess as to who this one is? I'll give you 10 seconds while I give a sip of water. Not that I need it, it's just a delay tactic. I see Bois, I see Arctic blue, I see Bois, Icarioides, yep, Bois. Yes, that is a Bois blue, Icarioides, Icaria Icarioides. Top side, really nice blue, not quite, well, I shouldn't say it's not stunning, but not quite super flashy or super iridescent. I've seen some of these are pretty dark blue as well, but just a nice even blue on these males. Locally common blue, that's one of the other common names for this blue is the common blue, even though it has a lot of species that can be a lot of subspecies, some of them which are threatened or endangered. But for the subspecies that are doing well, yeah, you get into a population, it can be quite common, probably one of the more common blues there. Let's see. So in general, so this is kind of a tough one because it is extremely variable, but you want to look for two things. This line of post-median spots here are usually what we call thickly ringed in white. So this is a particularly dramatic case. This one down here as well. Sometimes, okay, this is a pretty dramatically marked one, although certain subspecies can certainly get that, but that's pretty thick compared to another one we'll share later, which I'll tell you exactly that one. And then this crescent in the discal cell is usually very white. This one's kind of a little whitewash there. And that's going to be a pretty prominent mark. Now, other blues are going to have this, but they're going to have a couple of other ID characteristics. But this combo, these thickly white ringed post-median spots combined with that white crescent in the middle, that's a pretty distinctive feature, or a pretty, I shouldn't say distinctive, but consistent feature. So yeah, amount of white varies. Let's see.
David, there's a question about, and this may be a fisticuffs issue, but about whether Icaria is bowing now to Plumbagus.
Oh man, yeah, this is a fisticuffs one. But yeah, currently, so I'm going by the Butterflies of America website. That's, I mean, once again, contested on like, well, who are they? Who are they to say? I use them as my authority, and right now, Bois blue is in Icaria. But yeah, older sources will put this under Plebeius.
And John Pelham is also putting it under Icaria. Sorry.
Isn't it fun to hear people try for their first time?
Yeah.
So yeah, females are brown above with a little bit of blue scaling toward the base, and that comes in really handy for a particular butterfly that it's often confused with. So this is found in a wide variety of habitats, never really far from lupin though. I found them in sagebrush steppe, I found them in my subalpine survey sites, mid-elevation meadows, ponderosa pine forest, you name it. So long as there's lupin there, you got a strong possibility of finding Bois. They have a single brood every year, late spring to early summer, kind of depends on the local environment. Up at my subalpine spot, they fly late July, early August. And the hosts are strictly lupins.
Okay. Allison is commenting too that there's the Fender's blue in Oregon was just downlisted from endangered to threatened because they're doing better from restored habitat and reintroduction over the past 30 years.
Fantastic news. The common blue is becoming more common. So I do say beware because there is a look-alike species here. Anyone know what this is? You will find it flying with Bois in eastern Washington, on the eastern slopes of the Cascades. And depending on the timing of the resource you're using, depending on the year published, going to have different names. Let's see, one in the chat. Semiluna, correct. So if you're using an older source, this is known as the sooty hair streak that has since been split into 2 species. Ours in Washington are called the half moon hair streak. Still the same bug we've been seeing all our lives, just now under a different name. This is also a lupin feeder, and it also often occurs in the same locations as Vaudeville, often flying with it. Now, they do have a look to them, but you see there is those thickly ringed post-median spots, and there is that white crescent in the center, just like Vaudeville. But if we back up, take a look at those Vaudevilles, take a look at that half moon. I remember having a conversation with John Bauman over in Spokane. He sent me numerous photos of half moon hair streaks saying, I think these are all half moons. And I said, yes, you are correct. I think these are all semilunas. And he says, okay, but I don't know why. So his subconscious was picking up on the difference. His subconscious was saying, that's not a Vaudeville. I think it's semiluna. He couldn't explain it. The only thing I can say is that they tend to be a bit duskier, kind of more of a grainy brown ventral hind wing. If you get a good shot of their dorsal, which they're a hair streak, so they show you the top sides, but there is no blue whatsoever on these. They look like a female Vaudeville, but if you get a look at the top side, the female Vaudevilles will have at least some blue, whereas the half moon has none whatsoever. But that hind wing, to me, the best description I can give is very grainy, a grainy brown, versus kind of the more clean Vaudeville there.
So what makes that a hair streak in 25 words or less?
Ask the experts.
Yeah.
See, 3 words. So we do have a lot of hair streaks that actually don't have the hairs, don't have the tails.
Okay.
Tailless hair streaks. So you think of like the elephants, your coral hair streak, your bear's hair streaks. They're all hair streaks, but none of them got tails. Here's a tiny one.
Western pygmy.
There you go. So this one I actually photographed down in southern Idaho near Nampa, and they get up to Nampa somewhat regularly, at least more regularly than Washington. But yeah, that was my first encounter with them, and someone telling me they were tiny could not have prepared me for how tiny they actually are. I mean, that's my thumb, and I don't have particularly big hands, so tiny little suckers. So generally brown above. I mean, it's a blue, but it's not really that blue. There's some blue down by the base. Otherwise, you got on the underside kind of this striated gray brown with these large black marginal spots with a little bit of shiny scaling within them, but really nothing else you're going to confuse that with, provided you can actually see the tiny thing. So this is a rare migrant to Washington. Last year, we kind of talking about, for those of you who were a little late signing in, last year was a big year for Washington, where we actually had a strong migration of these blues down to Southeast Washington. I got skunked. I tried to get one of my West Side friends to go see it for the first time, and we checked a number of spots north of the record trying to find it. Didn't find it, but zeros are still good data, so at least we checked, and we can say, okay, well, they weren't there. Bummer. Still good data. Did find her first wild monarch though, so that was good. But since they're migrants, you primarily see them in fall, so they need time to get up here. In other words, big fan of rabbit brush. So if they get up here, you'll often find them nectaring, maybe right next to a giant monarch or something like that, which is kind of crazy to think about. But they also move, maybe not to the same degree of precision as the monarch does, but they still move vast distances just like the monarch. So crazy that little package can do the same thing as that 4-inch wingspan monarch. Larvae feed on a variety of weedy forbs, but Russian thistle seems to be a favorite, or at least that's the ones that we were seeing this migration mostly hanging around was in the Russian thistles. Kind of a nasty plant really to work with. But this is definitely worth a look, especially if you live more south, if you live Oregon, if you live California, if you live Southern Idaho. Us in Washington, we'll be crossing our fingers for this to happen more often, but don't hold your breath. Oh, God, that's bright. That is a bright blue. Okay, play acting is over. This is a really brilliant blue. And dorsal sides are nice once again, but let's show you the underside of this same blue. Any guesses as to who that is? You got 10 seconds. We got silvery, ligdamus. All correct. Who's got 100% so far? You can give the next talk. So yes, this is the silvery blue Glaucopsyche ligdamus. Quite a long name for such a short common name, but yeah, that's one of the big things. This is a blue you can, I shouldn't say 100% tell from the top side, but you get a really nice clue from the top side. That is a silvery blue. So they have a single generation every year, generally spring, early summer. They're among the first butterflies to fly. When WABA traditionally went our late March trip out to find the Sheridan's green hair streak, it was not uncommon to see the first male silvery blues flying out there as well, although often it would be Sheridan's hair streak and spring whites. But if you were there a little bit later, you'd find silvery blues flying among them. These are noted by having a single row of post-median spots. And you might be saying, like, what do you mean a row? So these post-median spots, the same ones that Waterval has, that's kind of your single row there, although it's a very wavy row. I get it. I get the complaint there. But pretty much just a single row, maybe the occasional spot here and there, but otherwise, it's a very clean marked butterfly on the bottom. It's found in a variety of habitats, sage steppe to subalpine, both sides of the Cascades. I mean, this particular individual was photographed at Magnuson Park in Seattle. Much more common east side and at elevation. Hosts are lupins, vetches, variety of other legumes. Is there anything else I want to say about silvery? I mean, the females aren't silvery, I'll tell you that much. Females are largely brown with a smattering of blue scaling, but once again, still the same ventral. Let's see, Regina says we should see silveries at Mima Mounds or Mima Mounds or whatever you want to call it. Yeah, you should. Regina, what date are you going to Mima at? What date is the Mima Mounds trip? Yeah, you get out of urban zones, you should find silvery blue with some regularity.
So
David, it's I'm sorry, it's a to be determined trip.
Oh, it's one of, okay. But
Yeah.
what time frame?
Mid-May.
Oh yeah, you should see silvery blues there. Ah, this is a nice one. Although of course I'm going to be saying that a lot this presentation. This is one that actually a lot of people it's on their bucket list. It's on their to search for list. And surprisingly, it's a close relative of the silvery blue, despite how dramatically marked this one is compared to the simple silvery. But they're in the same genus, and they're the only two in the gene. What's that?
Piasis?
Yes. Piasis? Piasis? So easy to say that when you're just reading it rather than, now I got to say it to everyone. So this might not surprise anybody, but this is the, oh, I got to click back in my presentation. This might not surprise anybody, but this is the arrowhead blue. I mean, come on, that's a great name. I love common names like that. I know I've said this story before, but I took a bird trip with my students, with a class of my students. And I handed them all out a checklist of all the birds we'd seen on our trips, and there's a bird that flew by and said, "Hey, what's that black bird with red wings?" And I said, "Look at your list." And they looked at their list and they went, It is not the red-winged blackbird. It's like, it is the red-winged blackbird. We don't try to fool you. So this is another case. Arrowhead blue. What a great name. And it's not just the arrowheads. You might think, well, duh, there's the arrowheads right there. Okay, well, this got a couple other clues as well. It's one of the larger ones. It can rival the blue copper in size. But if you just get, let's say, a photo of the top sides, here's a female. This is one of the ones where the female, yes, has less blue than the male, but she has quite a bit of blue for a female blue. But if you just get a dorsal side and you don't manage to get that ventral, the fringes are strongly checkered in this one. Now, there are other blues that are also checkered, but this is going to be a good clue to that. They have a late spring to early summer flight, so they'll be well after the silvery blues, but not quite late into the year. And they'll be found from like Columbia Basin to mountaintop, but kind of generally your canyons, sage steppe, open forest, dry subalpine. They are another lupin feeder. And I think, I'm going to have to research this one, but I think this one is a strong cannibal of the larvae are strong cannibals of other blues on their host plant. But I'm going to have to double check that. Don't quote me on it.
You're supposed to be on mute.
Who's supposed to be on mute?
I said you're supposed to be.
Anyway, but yeah, so this is a good one to put on your bucket list. If you haven't seen this one yet, check out a canyon called Number 2 Canyon in Wenatchee. Now, this has been maybe a decade or two since I've heard of this canyon, but I've heard that this is a canyon where this is actually a pretty common butterfly, whereas I'd say kind of like an Indra swallowtail. If you find 6 arrowhead blues in one day, you're doing pretty good. This is one that there'll be a giant puddle party of blues, and there might be one arrowhead in there, and that's my one one arrowhead for the day. Whereas I think it was Dave Nunnally who talked about Number 2 Canyon that says that's actually a common butterfly out there. So put that on your list. I don't know if they're still common out there or not. All right, here's another one. This is going to be in definitely a different spot than you find arrowhead. Feet get wet, where this butterfly is. Any guesses? You guys are good. Greenish blue, or Latin name Sapiolus. Once again, I can read it. Have never had to say it. So yeah, this was a butterfly that Pyle had the description, found where feet get wet. So this is the greenish blue. And yeah, generally lush, wet meadows, maybe some drier sites so long as clovers are abundant. But it's got much the same markings. This is one that's often kind of confused with bladderball, but you notice those spots, they're not thickly circled in white, especially if you get a little bit close to them, like they're thinly circled in white, but sometimes that background, that ground color of the ventral hindwing is so white as just plain washed out the white scaling. But the key feature you want to look for are these 2 rows of submarginal, or maybe you call them marginal spots, this pair of rows along the outside. That's a really good clue, as well as the habitat. Once again, identifying a butterfly might not all be about looks. You can take habitat into consideration. So am I in a wet meadow in summer? Probably a greenish blue. But this also has several disjunct populations. So we got your Cascades populations, you got your Eastern Washington populations, but it's got populations up at Hurricane Ridge in the Olympics, and then it's got some disjunct populations of different subspecies out on the coast of Oregon with kind of nothing in between. So very odd distribution with this one. One other cool thing I think about this butterfly is the sexes are dimorphic, which might not be much of a surprise in blues, given that between the males and females on the top side, but on the bottom side too, the females tend to be more of this kind of warm tan color. You can see in this mating pair here. The males are this much more chalky white, females more of this warm tan. The other point I should mention, there are individuals that will show a little bit of orange, just a tad bit of orange on the bottom side. Sometimes that's there, sometimes it's not. Here's the female on the top side though. Very brown, really no blue to speak of, little bit of orange on the dorsal hindwing there. And here you can kind of see the thinly circled black spots, thinly circled in white, much less so compared to a Baudival. Bob asked, are we recording this talk? We are recording it. There's one of my favorites. This is kind of one of my prizes on the Mountain Loop Highway. Any guesses on this one? Oh, 3. Okay. Oh, Arctic, Arctic, Arctic. Correct. You guys didn't need any refreshers. So this is another blue that I love this name because look at that blue color. That is an icy blue. That is a chilling blue. That is an Arctic blue. And then the bottom side, you might think, well, hey now, you said the Baudival had thickly white ringed black spots on the hindwing. Well, so does Arctic, but so much so that like the outer half of the ventral hindwing can almost appear completely white. So really thick hindwings, really thick white ring spots on the bottom, so much that it almost subsumes kind of the entire outer half of the wing there. So the male's got this icy blue above. Females, I think, are also very kind of stark, almost tundra-like. They're brown with these white rim discal cell spots on both of the wings. Let's see, yep, ventral hindwing with thick white scaling spots. These are high summer butterflies, so you think like mid-July to mid-August, although I've had a couple of my study sites kind of tail into September. So it depends on the elevation that you're at too. But this is a high elevation butterfly. I tend not to find these below 5,000 feet, except for one of my sites. One was puddling among some Anna's blues at a lakeside at about 3,200 feet, which is really low. But then of course I look up and there's a big old rocky cliff about 1,000 feet above me. I'm like, this male probably just descended from there to puddle, and he's going right back up. So mostly I associate this butterfly with ridges and scree, not so much meadows, although I think there's, and Melanie can correct me because I've never been to that, there's a, is it Corral Pass up in Mount Rainier that this butterfly
Yes.
is like that,
Yes.
like that's the highlight spot. Now, what is that place like? Is that meadows or is it kind of rocky scree?
So Dave Nunnally took us up there. He could probably speak to it very well, but where we saw them, it was in Rocky Scree, very high up.
Yeah.
Mount Rainier. So they really had it.
Yeah.
First
The
thing-
one of the spots on my study site, Mount Dickerman, has these, and they'll filter into the subalpine meadows up there, but I find them most often kind of at the rocky scree ledges, all that kind of stuff where their host plants grow. Host plants being mainly saxifrages, but a few other families of low herbaceous plants there. But yeah, I really like this butterfly a lot. I don't know, it's ours. It's a northern one.
So there's a couple questions here. One is it known as the northern blue Arctic?
I don't know if other older sites have called this one the northern blue, or if maybe it's known by the northern blue in other areas, because I think it's whole Arctic. I think they have it in Europe. But we do have a butterfly called the northern blue here that is not this butterfly.
And John says that northern blue is Clebagus edis.
Yes, and like I said, I just don't know if like regionally it's called something else, because like, for example, our Arctic skipper here is called the common checkered skipper in Britain, I think. But we have a common checkered skipper here that is not the same species. So once again, common names are great. They get people into it, but learn your Latin names, because that's where we're from here to Russia to Britain. We're all going to be in agreement on what this species is.
And Allison comments that in Oregon, they have the similar Sierra Nevada blue, but only from the California border up to the southern edge of Lane County, leaving a gap of about half the Oregon Cascades up to the Columbia River with none of these or any Arctic blues.
Yeah, the Sierra Nevada blue is nice. So if anyone's interested, I shouldn't be promoting myself on my own talk, but if you go to the Sierra Nevada Field Campus, they have a website. It's run by San Francisco State University, and they have a field campus up there. And I will be, I think, June 25th through 30th, leading a butterfly and moth workshop up there. And we do see Sierra Nevada blues on the trip. Now, maybe not that early, but there's a lot of nice California specialties down there. If you're a Washington person, if you're an Idaho person, you want to go see some new butterflies, that is a nice place to go see a variety of California species you just don't get up north. Sierra Nevada blue being one of them. So Sierra Nevada blue, not in our talk today because Washington don't have it. This ain't your talk. This is Washington's talk. But yeah, but relatives, I think they used to be considered the same species, but the Sierra Nevada blue uses a completely different host plant and does consistently look a bit different. All right, we're going to be seeing this one soon. I cannot wait. Any guesses? Regina says echo. All right, no other guesses. So yes, Regina's correct. This is your, oh, one more coming in. Yep, Stuart says echo. Correct. This is their echo azure. Maybe what our super old resource is called the spring azure, until someone did some work and figured out, oh, Celestrina's multiple species hidden in there. And our Western one being echo. Oh, gotta click back into my presentation. There we go. So Celestrina echo, our echo azure, males this brilliant blue, females with this thick black border, particularly on the forewing there. Chalky white below with these submarginal crescents enclosing these small dots. And Stuart Wetchler actually gave me a good pointer as far as remembering the Latin name. He said, think of these dots as a C and an O, co on the outside. There's your echo azure. The hosts are many species of woody shrubs. They often specialize by region, but this just kind of gives the butterfly a lot of flexibility to take advantage of what might have a good bud set in any given year. It is one of our earliest butterflies. It overwinters as a chrysalis. It cannot survive freezing temperatures in the adult stage. So when you see multiple of these males out, spring is here. There's no going back. This is better than any groundhog you can possibly ask for as far as, is spring here? I'm seeing echo blues. Yeah, spring's here. These are single or multi-brooded depending on the elevation. Down low, generally 2 generations. The big generations in spring, and then there's a smaller generation in summer, generally like late July through August or something like that. A lot of people don't notice them because they're not nearly as common. Generally inhabit shrubby habitats throughout the west, edges, things like that. Riparian in drier regions, and generally not found at higher elevations. Occasionally I have found them in my study sites up to like 4,500 feet, but I mean, it's as like singletons, not commonly. Speaking of higher elevations, one thing to note when trying to identify these things, let's say you find a blue, you swing at it with your net, you miss. Where does the butterfly fly? This I was taught by Pyle. These guys fly higher than our other blues, and it probably owes to them using shrubs and trees as their host. If you swing at this blue and you miss, it will fly up. Whereas at the same time, he was holding a different blue, one we haven't covered yet, so I won't give it away. But when he released it, it flew down and away. And all of our butterflies are going to fly down and away. This one will fly up. So that helps you with the idea if you miss and it flies up. Probably was an echo. Now here's a nice group of echoes, but hey now, that's something different there. Or is it just a smudgy echo? Nope, we got a different species there. So anyone want to guess what this one is? The one hiding amongst the echoes. Two people say Asher's blues. Kathy says Asher's. I don't know what you guys are talking about, Asher's. Clearly this is Lucia. What's an Asher? Oh yes, thank you, Caitlin, for finally clearing up the mystery there. So what we thought of, oh my God, I'm gonna get this totally wrong and Pelham's gonna hit me. We had three different identifications up here in the Pacific Northwest. We had echo, we had our nice cleanly marked echoes. We had our Lucia, which we thought was kind of an Ice Age relic holdover population that we only found in Yakima County. And then, oh, was it Dan Dunphy found another colony in, oh, not Bear Canyon. That doesn't matter. At the very
Yakima Canyon, and it was with Kathy Clark as well.
Yakima Canyon, okay, at the very southern edge of Ellensburg at a fishing spot. And that prompted Caitlin. Caitlin finally said, I'm going to go see this butterfly because I've never seen this butterfly yet. And that kind of catapulted her to study this butterfly for a year or so. She did the larva raising work. She looked at all the wing phenotypes, and then they sent it off to Nick Grisham to do the DNA work. And oh man, we got a new species here. This is our species. This is the Asher's blue, or I like the name Azure. I don't want to call it blue. Azure is such a good name. And especially since just keep it to the genus of Celestrinus. Like, okay, we got our Echo Azure and our Asher Azure. So I'm not giving up Azure. So it often flies with the Echo Azure, which makes things a little bit difficult, especially when these guys are lightly marked. But this used to be something we would just call a subspecies called migrescens. So we would think of them as like darkly marked Echo Azures. So in Eastern Washington, we would call them a subspecies where a fair number of these would be darkly marked. Now we're pretty much saying these are all Ashers. So what's the difference other than that big black spotch there? Because sometimes it can take up a large portion of the wing, sometimes a moderate amount, and sometimes very lightly marked to the point of like, wait, is that Ashers or not? In that case, there's two things I want you to look for. These crescents, the CO, the CO, are often at least some smudginess going on there. You look back at the echoes, those are nice, crisp, clean, dainty crescents. Whereas those are some pretty smudgy ones up here. Not quite dainty, a bit bold. And that blue on the top is really more silvery than that, what did Pyle call it? A periwinkle blue? I don't know. I feel like butterflies would be good on those paint splotch tests or whatever. So those three combos will help. Caitlin said something about seeing white arrowheads in here, which depending on the individual you can see, but I'd find that to be somewhat inconsistent. And then these fringes are consistently checkered as well compared to, you see, those are checkered compared to the Echo. Although you can make an argument for some checkered there, but this is strongly checkered compared to the Echo. So primarily eastern slopes of the Cascades and riparian canyons, although we do have records of them up at Snoqualmie Pass, at, kind of moderate elevations. And then of course, I had to go and find a western population up on the Mount Loop Highway. Thank you, David, for throwing another kink into the wrench. They were flying right next to Echo, probably Echo outnumbering these guys about 3-1, but they were definitely there. I caught a series of specimens just to quote unquote prove it. And these guys primarily feed on red oyster dogwood. I'm not sure if we've established any other hosts. I don't think we have. Maybe that's the only host that we know of at the moment. But yeah, this is a new butterfly for Pacific Northwest, Asher's azure. We do not have Lucia, so strike that off there. And as far as I know, nigrescens is no more for the moment. I think there's still kind of a little up in the air as far as like, we don't quite know what to do with nigrescens yet. And there's some of these individuals down in Oregon that are kind of key to figuring out what to do with the rest of this genus up in Washington. So Asher's azure, see, we're still finding new things. All right, one more guess while I take a water. I do need water after that round. Who's this one? Regina says gray hair streak. Chrissy says a tailed blue. Kathy says a western tailed. Daniel says western tailed blue. I'm sorry, guys, I included a hair streak in here. I mean, there's those tails right there. Clearly a hair streak, so. I'm gonna go with Regina's response. Clearly a hair streak. What? A tailed blue? There's tailed blues? All right, bad acting aside. Yeah, so we have two different species of tailed blues in Washington. And yeah, they can kind of fool you for hair streaks if you forget that we have tailed blues, which occasionally I do. Anyone ever heard of this thing called convergent evolution? If something works, it's likely to evolve more than once. So we've got swallowtails that evolved tails, we've got hair streaks that have evolved tails, and we've got a couple of blues that are evolving tails, and these little markings down there. The idea, although this hasn't really been tested, is that it kind of looks like false eyes and antenna to lure a predator's attack away from the vital end. Although, like I said, I don't think anyone's actually really done the tests on this. But yeah, so this is the western tailed blue, the more common of our two tailed blues in Washington. There are several characters you can use to distinguish this from the eastern tailed blue, although there are some spots where they get remarkably similar, like frustratingly similar. So in general, the male western tail is going to have no orange above, unless you get down to like the Southwest states, then they start to get orange above. But up here, don't have to worry about that. The female has more blue above than the eastern tail blue, and the bottom side is much more lightly marked with very little orange down here, whereas the eastern tail has much more orange on average. These are generally at higher elevation than eastern tailed blues, although they do occur in many habitats. They do co-occur in many habitats, but you can use habitat to a degree. Western tailed blues are much more likely to inhabit kind of these undisturbed habitats like meadows, stream sides, clearings, and usually there's at least somewhat of a moist component going on there. They are double brooded. Maybe this is conditional on what the year's doing, maybe elevation. The hosts are a number of different vetches, peas, loco weeds, other legumes. But here's your eastern tail. So notice that the males have a little bit of orange above there. I mean, it's small, but it's there. Females have little to no blue above compared to the female western. And notice on the ventral, much more orange usually than westerns, and they tend to be a bit more heavily marked. So notice the marking of the black spots. Let's go back to the western tail. That's a lightly marked butterfly, and there's your much more heavily marked eastern tail. These generally are found at more disturbed sites than western, maybe some agricultural settings, and generally at lower elevations. Although I have definitely found them in riparian spots that are undisturbed and flying with western tails. Problem here, as you get up to Spokane, the western tails start getting a lot more orange on them. and they start looking a lot like eastern tails, and they co-occur. It gets real frustrating real fast. Number of generations for this blue kind of depend on the elevation, and the hosts are flowers and seeds of many different kinds of legumes, but so still legumes. So I wouldn't be a good teacher if I didn't put you through some kind of a test, right? So you can feel free to put this in the chat box. You can feel free to keep your answers to yourself if you'd like. Which one's the eastern tail? So here's two females. Which one's eastern tailed? Which one's western tailed? For those of you playing at home, the left one is the western tail. Right one is the eastern tail. Remember, the eastern tail has very little, if any, blue on the top, whereas the western female has much more blue. Although remember, variation is the rule. So you might find some western females that have very little blue. Now let's look at the ventral. Which one's the eastern tail versus the western tail? So for those of you playing at home, on the left there is the eastern tail, much more orange, much more strongly marked. This is a really nicely marked eastern tail compared to your typical western blue there on the right. Once again, for those of you living in Spokane, very sorry, your westerns are going to look much more like easterns. Now, moving on to a completely different group, a fun group, a frustrating group. It gets harder from here. We move to the group of blues that have a lot of orange on the bottom side of the wing. And these come in three distinct groups. We have our group that has the aurora, however you want to pronounce it, on the ventral hind wing only, and no scintillae. The scintillae are these brilliantly spangled scales just outside of those aurorae. So the Euphaloides has these orange aurorae, no scintillae. Ichoricia, orange only on the ventral hind wing with scintillae. Plebeius is going to have orange on both the ventral, on the ventral, but both the forewing and hind wing with scintillae. So think of it as dividing into these three subgroups and start from there. Do I see scintillae? Do I not? Is the orange restricted to only the hind wing, or is it on both wings? So let's get into the first group, the Euphaloides. These are known as the buckwheat blues. As you might guess, their hosts are buckwheats. These are generally Eastern Washington. I shouldn't say generally Eastern Washington. They are Eastern Washington. I know of no Western Washington buckwheat blue. Generally more arid landscapes where their buckwheat plants grow, from lower basins to higher elevation mountain slopes, and they are single brooded. So in general, we used to think of, this was a while ago, we used to think that we only had two species of Euphaloides blues, the dotted versus the square spotted. And this is kind of how they fell into the two different categories here. Well, now we know the situation's much more complicated. We have several species in the dotted blue group and several species in the spotted blue group. Males, generally somewhat thick black borders and much darker blue than the average, but I'd say almost like a navy blue. Although, of course, as soon as you take a picture of that blue with a flash, it looks very, very blue. But in the field, you'll notice that's a very dark blue. Females, brown with the orange aurorae on the top of the dorsal hind wing there. Now, this is the worst situation you can find for identifying a Euphaloides in Washington. You find a Euphaloides puddling, or someone brings you a Euphaloides in a jar. So I caught this on the wing. Uh-oh. The best way to identify Euphaloides is when they're on their food plants, on their host plants, the buckwheats. Because these butterflies are very strict as to, I shouldn't say very strict, but they're often associated with particular buckwheats. And if you know your buckwheats, learn your buckwheats, you have a much better chance of identifying your Euphaloides. Now, this is where the last image I'm going to see for like maybe half a dozen of you is going to be your fist going up to the camera as you punch the screen. Currently, there are only two species of Euphylloides recognized in Washington. There are going to be some that disagree vehemently. We have so I'll start with the Columbia blue. This used to be the dotted blue or the Pacific dotted blue. And notice that the orange luneals, and this goes for dotted blues in general, are often separated. So you have each of these in their little orange crescents separated from each other. Nice separated crescents. And the ventral hindwing is much cleaner, kind of less dusky. The spots are smaller, more dainty overall. Only one generation year, but different populations are on different hosts, so it might seem like it has a long flight time. But for those that feed on elatum, might fly a little bit later than those that feed on compositum, but there is some overlap, so they're still trading genes back and forth. Occasionally, they'll also feed on nudum. This is where some of the confusions come in, because some will say that we do have Pacific dotted blues in Washington, this species Enoptes, and they feed on nudum. And there's some contention that we do not actually have Enoptes here. We have Columbiae that occasionally feeds on nudum, Eriogonum nudum. Now, I mean, you can see why it's called nudum, the naked buckwheat. I can barely see the buckwheat in that picture there, but it's there. And if you're a blue feeding on that buckwheat, on those flowers, you're going to be fairly small. So maybe that confounds the issue that maybe Columbiae on nudum just turn out to be a little small. Versus our other species of buckwheat blue, our other species of Euphylloides in Washington, the Cascadia blue, new species. It actually has been officially described. We used to describe it as Euphylloides on Heraculoides, which is one of its Eriogonum host plants, Eriogonum Heraculoides. Now it's just Euphylloides Heraculoides. So the orange lunules are expanded and often fused to form an orange band here. Much, much harder to distinguish each individual aurorae there. And the ventral hindwing just kind of duskier overall, kind of much more spattering of black scaling. And those black spots are much larger and more blocky. So if we go back to the side by side, now you can kind of see those spots on the spotted side, much more blocky than those on the dotted side. You can see where you got the dotted versus spotted name. Those orange lunules, much expanded, kind of more of a band versus the dotted side, kind of more individual crescents. Much cleaner look over here than the more dusky spotted. So luckily in Washington, currently, we only recognize the two. Now, the Cascadia blue feeds on more than just Heraculoides. It'll also use Douglasii, and it'll often switch from each population might feed on both of them, switching back and forth pretty readily. But we used to think we had this one called the Glaucon blue, which goes up to, I think it's California, maybe there's some populations in Oregon, but they feed on umbulatum, which is a much more kind of lower matted, sprawling buckwheat. And we have populations that are using umbulatum, hence we said, hey, we have Glaucon. But we have these populations kind of dispersed pretty randomly throughout Washington. And they're at a bit higher elevation where Heraculoides buckwheats just aren't there. So this seems to be more of one species, the Cascadia blue. As it expands, it takes advantage of other buckwheats. So if you go up Icicle Creek Canyon outside of Leavenworth, you find these Cascadia blues feeding on umbulatum. Now there's a couple other populations that feed on Spirocephalum and Thymoides. And I don't want to promise anything because I'm not a Euphylloides expert, but we're kind of working out, is this a different species? Because they do consistently look different, or they look unique. But could this be a different species entirely, or could it be maybe a phenotype triggered by the host plant that it's using? I really don't know. But there are populations of what we are currently calling Cascadia, the Heraculoides, feeding on Thymoides and Spirocephalum. So more news to come. Once again, currently we recognize 2 species. This may change, and I put an asterisk next to may and say probably will definitely change. Probably will definitely. All right, now we get into 2 much harder group. Now we get into 2 much harder species. We're getting to the tough spots now. Acmon versus lupin. This is like banging your head against the wall stuff. Before we get into this, I see we got some stuff in the chat, so I want to look at that before I get too far into these. Okay, that's just about the key. Got it. So here we have male Acmon on the left versus male lupin blue on the right. Down here are the females. You see the females really vary on how much blue they have. So the blue is not really a good indication on females, like it is, say, with the tailed blues. So Acmon blue, I think of about 5 different clues I use to distinguish these blues from each other. Sometimes I get 4 out of 5, and that'll have to be good enough. And sometimes we have to be comfortable with the answer, I don't know. That's a legitimate answer. So Acmon blue, first one we'll go over. So you look at the aurorae on the top. This is one of the groups that has, now these are Ichoricia, just to let you know. So the same genus as the Bois blue and the greenish blue currently. But these are the only 2 species that the males have orange on the top side, at least in our state. So if you see a male dorsally with its wings open and you see that orange, you know, hey, it's one of the 2. So nothing says you have to get down to species right away. Start with the groups, work your way downward. So the orange aurorae on the dorsal hind wing, I say in the ventral hind wing, I'm not sure if I meant that. But on the dorsal, these are kind of more pinkish, more of a pinkish orange. And the dorsal, oh, aurorae on, there's a typo. I need to revise that. And these aurorae meet the blue scaling directly. And you'll see what I mean by, well, why does that matter? So this blue tends to be a more pale, brighter blue above than the lupin blue. And the black borders tend to be much thinner. Remember, up here, tends to be. These are also multi-brooded, especially at lower elevations and farther south. And this really helps when you consider what time of year are we finding this. Are you finding it in April? Probably in Acmon. Are you finding it in August or September? Probably in Acmon. And they're found in virtually every open habitat along the West Coast. These are travelers. They're an adventitious weedy species. So you'll find them down in the Columbia Basin. You'll find them up at some elevation. Was it Pelham saying in a key, he would say, is there a discarded couch or old junk car next to you? It's an Acmon. So they'll take these kind of weedy wasteland places as well. They have a variety of different hosts, including buckwheats, legumes, and probably many, many others. Versus, so keep all that in mind. Let's go on to the lupin blue. By the way, lupin blue, terrible common name, has nothing to do with lupins. Its hosts are buckwheats. I do not know the history as to why they're called lupin blues, but hey, the name stuck, so there it is. It's still lupini as far as its Latin name. Oh boy. So the dorsal aurorae, so once again on the dorsal hindwing, are more orange. They're more of a red orange than that pinkish orange of Acmon. And they're separated by the blue scaling by a thin layer of black scaling. Sometimes it takes some imagination to see this. And the blue tends to be a deeper blue than Acmon, and they tend to have wider black borders. Like this one right here is like just a quintessential lupin blue right there. Now these are more of a mountainous species, although they will be down in some canyons, but they can get up to subalpines, arid hill stuff, and they have a midsummer generation. So you tend to find these in June, maybe a little bit later the higher up you go. So if you find a blue in June, it could be either or. Once again, you use timing though to help. If you find it in April, when I was living in Walla Walla and I found fresh males in mid-April, okay, those are going to be Akmon. Versus you find one in August, that's going to be Akmon. You find some in a mountain area in June, it could be either or. So start looking at these little characteristics here. So if you go back, now that we've gone over those physical characteristics, we go back to the side-by-side comparison. So you see this is a much more faded kind of, yeah, pinkish orange meets the blue directly, much more red orange on the lupin blue, and that thin layer of black scaling in between. Much thicker border on average than the much thinner black border on the male Akmon. Females, oh boy, I haven't heard of any real key features with the females yet. Once again, timing and habitat will help. So once again, a quiz on the toughest group, on one of the toughest pairs of butterflies you can find in Washington. So you can play at home if you'd like. What do we call number one? Regina says Akmon. I would agree with her. What about number two? Anyone want to hazard a guess to number two? Once again, how deep you call a blue, the blue color depends on sometimes the flash, the angle of lighting, the length of exposure, all that kind of stuff. So let's see. Regina says lupin, Chrissy says lupin. I would agree. I'd say that's a pretty nice lupin. Here's number three. It's a female. That doesn't really help. What if I told you I found this in Swakane Canyon in late August? You're gonna go, oh, late August. So yeah, that's probably an Akmon. I would guess so too, because you would find lupin blues in Swakane Canyon, but more around June, more around late June. So this is well outside of the regular flight period. This is most probably an Akmon. What about number four? Any guesses on number four? As far as timing, let's say this one was late June, early July in Swakane Canyon. I noticed that the chat box is eerily silent. I see lupin question mark. Yeah, I included this one because this is going to be those ones that if someone sent me a photo with the description I just mentioned, that's going to be an I don't know, because maybe some could say that, you know, God, is that black scaling or is that where? Is that is more orange, but that black border, that's neither really thick nor thin. So this would be one of those, it's like, it's so in the middle. I don't know what that is. Let's see. Christy says, Steve Kohler says that all of ours in Montana are lupin, and that we do not have Akmon here. Makes it easy for us. Yeah, but you're also missing Akmon. So, you know, can you really say that makes you happy that you don't have Akmon? Ooh, now there's something interesting. That's a lot more colorful. Let's see. Tyler says, my understanding is that BC only has Akmon too. Others on the call may be able to confirm. I will defer to the experts there. My impression is that Akmon doesn't overwinter too far north, and so it kind of has to expand every year. So I don't know if it gets up to BC. So this is part of that third group that I talked about, remember? We've got the scintilla, not that clear in this photo, but we have orange on both the hind wing and the forewing. So this is that third group. Oh, gotta click back in there again. I'm gonna zoom backwards really quick. There's that third group, the plebeius. Plebigists. Ah. Zooming back. So this is, oh, Christy says Melissa. Oh, Pelham says no records of Akmon for BC. Oh, interesting. Time to start dissecting them up there. So Christy says Melissa / northern on this one. I'd call this the Melissa blue, especially given where this was Swakane Canyon in August. So that's going to be Melissa. Now, sorry, I'm getting discombobulated around here. So the best term I heard for describing the amount of orange on Melissa was screaming orange. So especially on the hind wing and especially on females, because the females get orange on the top side on both wings as well. And then the amount of blue on the females varies quite a bit. I mean, wouldn't this be a spectacular creature to see, wouldn't it? With between all the blue and all the orange. You look at this female down here on the lower right, and that is just screaming orange there. Let's see. Yep, amount of blue varies on the females. Multi-brooded, so they'll fly from, I think, like April to September. Usually 2 broods, maybe 3 if the season allows for it. Eastern Washington, generally lower elevation, kind of drier, more disturbed. Sage steppe, agriculture, roadside ditches. Not really what you might consider pristine, so to speak. Well, I guess sage steppe can be pristine while not seeming pristine. They feed on a variety of legumes, but this species particularly benefited from all of our alfalfa plantings. Does like to lay its eggs on that. Versus the other butterfly this does get confused with, especially in northeastern Washington, is the northern blue. So this is Plebius itis. This is the one that someone said, hey, is Arctic blue considered northern blue? Maybe by region, but here in Washington, our northern blue is Plebius itis, and it's restricted in Washington to northeast and southeast Washington. And these are mostly at higher elevations, although Melissa can sometimes overlap. One thing that Pelham taught me, northern blues have, on average, once again, everything is on average here, variation is the rule, so keep that in mind. Male northerns on average have a thicker black border than the average Melissa does. So that might help. In addition, oh, why didn't I mention the orange? Yeah, so you do have orange, prominent orange on the ventral hindwing and the ventral forewing, but it's not screaming orange as the average Melissa is. And then same thing with the females. Females will have orange, but maybe not as much orange. Timing can also help. So if you find some worn, kind of moderately oranged blues up at, let's say, a classic spot is Moses Meadows in north central Washington, and as you descend, you get some really bright males that have a lot of orange or have maybe moderate orange. Well, what's this bright male doing at the tail end of this blue population? You're probably looking at an overlap zone where a new generation of Melissa blue is coming out, while the northern blue, the single generation northern blue, is reaching the end of its flight. So timing can help there. Kind of consider what stages the males are at, what timing the females are at, all that kind of stuff. They feed on a number of legumes, but lupins and vetches are kind of the favorite in Washington. Once again, another look at northern versus Melissa there. That's looking a little different. So now we're in the Cascades. This is the one, if you are a summer hiker in the Cascades, if you're a summer hiker around Rainier, or any of these classic summertime trails, if you're a Seattleite, this is probably the most common blue you see in the mountains. This is Anna. So the first thing you might notice, yes, there is orange on the hind wing. God, maybe there's orange on the forewing. Yeah, there's scintilla, but kind of barely. And that's kind of the key to Anna's. It's a much more muted Plebius than the northern or the Melissa. So it varies. This is probably one of the more striking Anna's blues I've seen as far as the amount of orange, versus this might be kind of your more average. And this is unfortunately very common as well. Kind of these very worn looking blues with just a smidge of orange and a smidge of scintilla. Someone might mistake this understandably for a bois de val because there's a white crescent there in the middle, but generally these are much later flying than other blues. So they kind of first come out around August or so, and they go well into September. Whereas they'll fly with Baudival, but Baudival are usually on their way out as these guys are reaching peak abundance. The bluish scaling at the base of the ventral hindwing may also be a really good clue. So this flush of blue scaling, although this one you can see doesn't really have much of it. These are usually at higher elevations than Melissa. So Cascades, Melissa will go up the slope with East Cascades, but I don't think it overlaps too much in Washington. I know there are populations in California where they have hybridized, but I don't think that occurs in Washington, though I may be just out of the loop on that one. Host plants are lupins, other legumes on occasion. We got a single summer brood, and yep, high elevation meadows and the Cascades and Olympics. Once again, the most abundant blue butterfly for summertime hikers in the Cascades. Females, much more reduced orange on average, although some individuals do get screaming orange, but that shouldn't be an issue when you consider where you are. Kind of just some other images of these blues. You see how washed out these can get. I mean, yeah, like that's a good Anna's right there. You got some orange, some centilla, but like that male there is just, it is just washed out. But that flush of blue scaling on the bottom might come to your rescue. So that is all 17 of our blues here. And so I did want to mention, although it has been mentioned before, I call it a new tool, because I say new tool because I think I first created this back in 2011. Oh God, over 10 years ago. What am I doing with my life? Anyway, existential problems later. I developed this thing called a dichotomous key to the blues of Washington State, because there are a number of species here. They're small. I can make all this stuff look real easy because I've blown it up to the size of, a birdwing butterfly or something like that. But then you get out there, they're this small. They're an inch wingspan. The pygmy blues are as small as your pinky nail or something like that. And all of a sudden, wait, I'm looking for that spot, that tiny little spot up there in this cornucopia of color and movement and stuff like that. That gets really difficult really fast. So I wanted to make some kind of tool for photographers, watchers, collectors, whoever, to kind of whittle this down. So if you go to Washington Butterfly Association, go to resources, learning aids, and there's my key to the blues. You see it is now updated March 1st, 2023. Although Carolyn has provided the link in the chat. Thank you very much, Carolyn, for posting that so quickly. And so why do we need, I need to update this because it's not new anymore. It's over 10 years old, but why do I keep updating it? I guess I should say, well, we've had recent taxonomic changes, new discoveries. We got a new species. Our understanding of euphalloides is getting better, maybe. We have maybe some new arrivals might come into Washington. And this will just help a beginner or intermediate gain some more confidence in identifying their blues. So for those of you who haven't used a dichotomous key before, basically it's a system of identifying objects. The classic one is like your flora of the Pacific Northwest. Generally like these paired choices. So it'll give some kind of separation of characteristics that lead progressively to identification of the object, which is often just the only remaining choice. Think of it as like, I heard a good analogy is like one of those choose your own adventure kind of stories. So for example, we're not going to go through the whole thing, but just one little example of how this might work. So let's begin. So the blue creature being viewed in Washington has wings. Well, then you would go to the dichotomous key of blues for the blues of Washington State. But then choice number 2, the creature being viewed does not have wings, is usually found in Las Vegas and most commonly found in groups of 3. Well, then we go to the next slide. And that, ladies and gentlemen, is my last bad joke of the presentation. Thank you for joining me tonight. and may spring please come soon so we can see some echosaurs flying for once. I'll take any other questions, and then, yeah, we can get some of the experts chiming in with any taxonomic questions that I may not be expert enough or confident enough to get.
So everyone's free to unmute. This is open discussion. Fire away. I did have one thing I just wanted to say, that Anna's blue pair that you showed, they don't even look like the same species.
They don't.
They look so different.
See my kids.
What's
What's that?
John?
I said you ought to see my kids, you know. The ink is black, the paper is white, you know. It's like that's the way it goes. I want to get in my licks, but well I can. So we have like really long-term surveys at Reeser Creek, and a reasonably long and growing survey at the.
We're eating.
What's that?
Oh.
Pretty decent survey from the Belline Rao Ranch. Cheryl Belline is, I believe, online, and we're able to document that the Acmons are first to emerge, like David said, and the lupins come after them, and then the second brood of the Acmons come after the lupins. And so, like, this is actually pretty well demonstrated, and we can actually show that the lupins are right in the middle of the other two broods. It's harder if you're visiting someplace to know if you're on the other side of one thing or another, but when you have a long-term study at one place, it all becomes pretty evident. But I'm with David. There's individuals that if you have a photograph or something like that, it's hard to call.
So David, when you say high elevation, I find Melissa blues up in Mount Spokane, just puddling all over along the road for people driving over the top of them. It's my favorite butterfly over here. How high?
Melissa blue on the top of Mount Spokane?
Yes, not quite to the top, but almost up to the top.
And that might be northern.
Okay.
So we have northern blue recorded from Mount Spokane, so.
Okay.
I have a question about the arrowhead blues. You mentioned, David, that you don't see very many of them, and even if you do see them, you just see onesies or twosies. It seems extraordinary that they can continue as a population if you see them so infrequently.
We can think of a number of our butterflies that do that. They have ways of finding each other. But Pelham can probably speak more along the lines to that than I can.
Yeah, it's really pretty easy. Butterfly watchers and collectors are very poor predators. We're not really good at finding the things that we're looking for. Things that are low density might actually be represented over a large area by higher densities. We might be in the wrong place where they're not abundant. There's all kinds of factors that play in. This was especially evident after 1980 eruption of St. Helens, when we had a couple of our favorite places, heavily dusted with ash. And for years afterwards, a couple of the species that we had recorded the spring before or the spring of the election, but before the eruption. And it took 2 or 3 years for these things to recover, despite the fact that we visited there all of those years and didn't see them. So our ability to see things is, limited, I guess I could say.
So, Matthew, do you want to unmute and comment?
Sure. I guess I was just lurking. I don't know. Yeah, just because we're on the ground doesn't really mean butterflies are going to be on the ground at that particular time. So I think there's one species I think David had mentioned in the past. I think you'd spoken with John on it as well, that it really spends more time in the canopy. And then I've also heard some people talk about activity that increases like at the dusk hours or early in the morning. And so there's a lot of variations. But with that said, whenever you go into the field, if you're not necessarily finding something at that particular time and place, a 0 is still good data
Yes.
until you get something else. So, all right.
That it?
But yeah,
But not
it
very
is,
satisfying.
but it is interesting though that you would have number 2 canyon is a place where they get common. So I don't know what's going on there or not, but I have yet to beat a number 2 canyon myself, but this is off of Dave Nunnally's recommendation from a while ago. So I don't know how true that still holds.
This is Dave Nunnally. Number 2 canyon is loaded with Lupinus sericius, the silky, is it silky lupin?
Yeah.
And that's one of the primary plants for the arrowhead blue. I haven't been up there for a few years, but I gave the directions to another fellow who was up 2 or 3 years ago, and he said they're still all over the place up there.
Yeah, if arrowhead blues are still on your bucket list, go to number 2 canyon in Wenatchee.
That's hard to get to.
It is?
The road up is in just terrible shape.
Oh, we're
you
used
need-
to that in Washington.
No, this is terribler.
Oh, lovely.
Dave, I remember you remarking that like mid-May one year, you saw about 30 arrowheads at Swakane Canyon. Do you remember that?
Not specifically, but they can be reasonably common there, but not all the time. Usually you see, like David was saying in his presentation, you see 1 or 2 or 3, but occasionally you'll see more.
Yeah, that's what I've seen there. It's just a couple, but where we have seen a large population was at Spiva Butte. Lots of lupin there. John, you were there, and they were just so abundant. Given that I'd only seen 1 or 2, at a time, to see so many flying from plant to plant, males and females, it was a real treat. And unfortunately, the area has burned. I'm not sure, how it is recovering, but
Yeah, well, 2 things. We call that draw arrowhead draw. And second thing, burning might actually be really healthy for that. That particular species may, prosper, not that the other habitats won't, but I've been places, 2 or 3 times at the same place, and I've been there, not seen an arrowhead blue, and then seen maybe 30, 40, 50 on one occasion. So there's, population explosions can take place too, and they're random. You can't predict them, so if you're poking your nose in enough places where they could be, then you're likely at some point to come across a time when they're really common.
So Diane, do you want to comment on your experience in Oregon?
Say what?
Repeat your question.
Diane, if you're still on, do you want to comment on your experience in Oregon with the arrowhead blues?
Not me.
Looks like Diane may be gone.
Okay.
Oh, she's still here.
Okay, Diane, if you want to unmute and talk about your experience at Mount Ashland, that would be interesting.
Maybe doesn't have a microphone.
Okay, maybe. So she said that she has seen arrowhead blues on Mount Ashland in Oregon.
And they're also in the Ochagos and up in the Metolius. We get them on the counts every year.
So in abundance, or just 1s or 2s, or?
1s or 2s.
Okay. So the fisticuff issues, is that euphalotes? Is that the main issue of contention?
Euphalotes, and then if you look at the Labar pile book, there's the volcano blue, the Icaricia on Eriogonum pyralifolium. Is that the one?
Yeah, I can report on that. Nick just came back with the genomic sequences on those. They're lupini. There's no
Yeah.
question.
So that's so that was the I should say that was a contention. I don't know if anyone's going to continue to hold a contention, but yeah, Euphaloides is still the one that there's most contention around.
No, I only told you that, David, because Nunnally's on the line and he probably disagrees, you know?
Well, like I said, I predict that in 10 years we'll have a different answer.
So
So
David,
Steve Kohler in Montana put out a paper, was it 2 years ago? On the euphalotes, and I don't know if it covered Montana, just Montana or all over the place, but after reading that paper, I concluded I can't identify any of them because it was like they all had to be dissected. And so I don't know what to do with any of my pictures for them.
And that's where you continue to look at the Buckwheat Association, or be satisfied to saying, hey, it's a euphaloides. Or you can say, is it of the dotted or the spotted group? And maybe even some of those, you're like, I don't know, but that's fine.
All I could do with my pictures.
And get good at identifying buckwheats.
My buddy Steve described Heracleoides that David just covered. So he spread himself afar. That was an excellent paper, by the way. I just really like that. Steve is incredible. Do you know him?
Yeah, he is amazing. I know his son a lot better, who's odonate expert.
Oh yeah.
Yeah, Nate. He, and tiger beetles. I take tiger beetle pictures. I email them to Nate. Say, what is this? But yeah, his dad is. I've been wanting to get over to Steve's house and look at some stuff in his collection just to help myself with ID in the field. But, with the pandemic and everything, I had never got over there. And so, one of these days I may shoot him an email and see if he'd be willing to have me masked up or tested beforehand or something. As far as I know, he's doing okay. From when Nate said he spends a lot of time fishing when he isn't doing butterflies. So he's a busy guy. We keep wishing he's going to finish the new Butterflies of Montana, but I think he gets too distracted publishing and fishing, from what Nate says.
Well, I'm on him about the Butterflies of Montana all the time. So like if you add a note or two, then that would be appropriate. But we don't call it the Kohler Residence. It's the Kohler Museum.
Yeah, that's what I've heard. And I've talked to people who've been over there and they said it's just amazing. So he's a pretty modest guy for what he's been doing.
Montana's such a little state, it should be a piece of cake.
Yeah. Yeah, I'm actually hoping to get camping over on the eastern end of it where I did my graduate studies, because I haven't been over there in a long time, and just to see what's out there. But anyway, yeah, it's a lot of country, and we went up to the Beartooth Plateau this summer, and I'm still scratching my head over the fritillaries. So anybody, David, I'm ready for you to do a fritillary ID talk, even if it doesn't quite cover all of ours.
David, come on, David.
I think you're just going to scratch your head more after that.
Probably.
I tried one presentation thinking, I've got this, I've nailed this, I've got this so for beginners now. And I was met with just this deer in the headlights stare after my presentation, like, crap, I didn't do anything.
So Dave Nunnally, you didn't take the bait that David Drappers tossed out about the euphalotes and the buckwheats. Do you have any comments, or just didn't know if you were ready to go to fisticuffs?
I didn't catch all of that. Could you repeat?
I was just wondering if you had any comments in response to David Drappers about the euphalotes.
Well, now hold on. My opinion is not. I'm not ready to go to fisticuffs over my opinion. I'm listening to all the experts.
Well, Well, the euphalotes are really tough. John's, of course, been talking about a new species, the Sphaerocephalum and Timoides feeders, and that's probably going to be a new species, but that's not described yet.
Mhm.
You mean I'm gonna have to update the key again? Ugh.
Of course.
Yes.
One more comment if I can go on there. So just because I've never been to number 2 Canyon, I just quickly looked it up on Washington Trails Association. And yes, the dirt road when the pavement ends is everyone's saying is terrible, but there's a parking lot down at the bottom. So for those of you who can hike, who can walk the road up, there's a parking lot at the bottom at the end of the pavement. So you can go see arrowhead blues in abundance if you're willing to walk a mile up a dirt road.
Just a mile?
I think so.
Oh, it's not that-
Probably a little less than a mile. I've driven up to the about 3/4 of a mile or a mile above that really bad spot. The road takes a hard bend to the left, and there's an old road that goes straight with a gate across it, and that's where you go in for the arrowhead blues. But we've driven up there a few times, but every year it got worse until it got, even for a 4-wheel drive, it's almost impassable.
So, but the good news is there is a parking lot down low as long as you're up for a walk.
Yep.
Anyway, stop talking about arrowhead blues, I guess. But yeah, just to reiterate, I'm seeing people saying arrowheads at many locations in Southern Oregon, Mount Ashland. Oh, they're widespread. It's just if you want a good chance of finding a bunch of them, it seems like number 2 Canyon's the place.
Yeah.
Sounds like a good place for a flash field trip.
It might be. I'm planning my last year on this being my Mountain Loop Highway research because I'm feeling trapped. There's eastern species I haven't seen in years. So I'm tired of having my species list below 20. I get back out to east and get 30 plus.
And you didn't find the Johnson's hair streak along the mountain loop?
What's that? I did or didn't?
I was asking.
Oh, yeah, no, I got two locations. Two locations. I'm suspecting 2 more, but we'll see what this June brings.
Mhm.
Well, it is 7:45, folks. If anyone wants to dive out at this point, thank you all for joining.
Could I get an expert opinion on this what of all blue genus? I'm going to
Icaria versus Plebius.
Yeah, John, what do you think?
Well, as author of the catalog, I just think, I mean, there's no doubt about it. It's not Plebius. Okay. So like, it was like, yeah, it was about a decade ago when worldwide study of blues, broke it down genomically and morphologically. And of course, Nick Grisham since that time has considered there's no question that Icaria is a compact genus that includes some very different looking blues, including Icoroides and, like say Agmon. That now Plebius is at this time only has 3 species considered for the North America, but
okay, good. That saves
that my catalog is online. That could save you a lot of grief, I think.
Okay.
All right. I'll send you a link.
I think I have it. Thank you.
Oh.
Okay, any other comments or questions?
Doesn't feel like it, but spring's right around the corner.
I was just gonna say, David, you've just done great work in getting us all revved up and ready for spring. So thank you for that. Yeah, this is exciting.
You all are lucky. You get spring a little earlier than we do.
Oh,
And we've had 4 months of winter now. And as much as I like winter, I am ready for spring. And we got feet of snow out there still.
this is
You may not be seeing any butterflies in March.
Well, my husband and I are gonna get the camper and we're heading to southern Utah, hopefully
There you
in
go.
April, so we can thaw out.
this is. Oh, sorry. Go on, Dave.
I was just asking, where are you, Christy?
Missoula, Montana.
Oh, okay.
Yeah. Shout out to Jeff Thompson, my neighbor to the south in Idaho.
Jeff. His house must be a predator bird haven, because he's always putting things that all these hungry looking birds standing around waiting for him to feed them mice, I guess.
Well, we still have winter here in Idaho as well, but I expect to see some butterflies in March.
Yeah, we usually have some coming out in March too. This is right now, we have a lot of hard crusty snow in the valleys, so we'll see what happens. See if we can get it melted here pretty soon.
Jeff,
On a
what part
good-
of Idaho are you in?
I'm Lemhi County, just a straight line below Missoula, Montana, across the Continental Divide. We're about 4,000 feet here in the valley. On a good warm day in April, I'll go up and find some Compton tortoiseshells that have overwintered here.
So are you on the eastern side of the Rockies then?
No, we're on the western
On
side
the
of
western
the
side of
divide.
the Rockies, okay.
Yeah,
just
yeah.
west of the
We're
divide.
still Pacific Northwest,
Okay, just.
Yeah. just barely.
Yeah.
And you often post beautiful pictures of Indra swallowtails. Is that,
Yes, I think we're the Northwest capital for Indras. I just see dozens and dozens of them.
yes, I look
They're
forward.
probably our most predominant swallowtail.
Boy, this coming summer, Jeff, I gotta head down there and because I've never seen one, so I'm gonna have to get a hold of you and come down and look at them.
Between 1st of May and about June 15th, we can just get a pile of them.
Great.
Another field trip.
So Allison, do you want to weigh in?
I just wanted to thank David for this presentation, and especially between the Ackman blues and lupin blues. And we have one cascade count here in Oregon that we turn into the NABA 4th of July counts every year. And hopefully someone doesn't try to tease out what the lupin blue and Ackman blue populations look like, because every year, depending who's leading the counts, either we have a whole bunch of lupin and no Ackmans, or we have a whole bunch of Ackmans and no lupin blues.
Well, both could be true. I mean,
Yeah,
I've been places. If you get a delayed season, then both could be true. You could be there, and see one or the other, and not both.
that is true. And some year people put down both. But I think some of it's true and some of it is misidentification.
Well, hey,
Well, here in western Montana, the northern blues and Melissa blues, I mean, our northerns can have quite a bit of orange on them. And we all, I took over the butterfly count here years ago from Will Curling, and he was calling everything Melissas. And so we called them Melissas for years until Steve Kohler straightened us out. And now they're all northerns. So that was all misidentification.
John, tell us about the habitat for northerns and Melissas.
what did John Pelham do?
Tell them about the habitat differences for Melissa and northern.
Number one thing is the Melissa blue is kind of like a weedy butterfly. It's in the low elevations. It'll penetrate up, step canyons, like David was saying. They'll get up the roads on the way to places like Moses Meadows, but generally they're confined to the lowlands, and northerns are in boreal forest habitats, right? And he went over the main features. One thing that he could have maybe emphasized is that a lot of northern blue females will be almost absent any of the orange on the forewing. So like they could have orange crenulations on the dorsal hind wing, but very, very, very little orange on the forewing. And so Melissas are never like that. Melissas are always screaming. They're always, have a real profound, orange bands on both wings. But, it's a feel thing too. There's some Melissas that would be kind of confusing. And there's a lot of northern blues that could be confusing because unless you're like seeing a lot of them, if you see one or two, then you say, "Ah, looks like a Melissa's," and it could be tough, to call them. There's a lot of butterflies like that. In case somebody didn't know that. Some butterflies you just have to be. Okay, I saw Frit, right? I saw Melissa Northern or a Northern Melissa,
We're going to be having the conference up in Ponderay County this year. And the town of Ione is up in that area. And there's probably the brightest northern blues that I've seen are right in the town of Ione. I thought they were Melissa's for sure, but John convinced me that there's northerns. So that's a good place to look for some very strongly marked northerns.
Cool.
So the information about the conference is posted, I think it's posted on the website, isn't it, Carolyn?
Yes, there's some information about it, but there's a lot more to come about the conference. But
So
the basic, the dates and the where is there, but
So for
no
those of you-
details.
Go ahead.
Nothing about registering yet, but
So for people who are unfamiliar, it's a multi-day butterfly event in a location that, it's a particular location with lots of field trips and experts to help with figuring out what the butterflies are and where to go. So if anybody's interested, it's really just a fun time to hang out with other butterfly geeks and spend a few days running around.
What'd you say, butterfly geeks?
I'm talking to you.
I don't want to make any promises, but I think I might actually be able to make this one.
Oh, yay!
Good.
So we'll see. We'll see.
Oh, that would be really lovely, David.
Would be. Well, I'm not a member, but I guess the American Lepidoptera Society, they're going to be meeting in Billings this summer. So I'm thinking about joining and going down to their meetings.
Oh, you should definitely do that. It's a great time. And everyone there is like, they're just like us. They're just local folks, but they come from all around the country. I'm going to be there.
You're
Good.
going to be there, and Caitlin's going to be talking about the new species, isn't she?
I don't know if she's committed to that.
Oh, okay. I'm sorry. I thought she had.
Well, I do know if I say she's committed to that, she's going to be angry with me.
Anyway, she's going to that conference too, though, isn't she?
Yeah. Caitlin and Ben Mouse and I are all going. That's going to be a fun trip. I get to watch.
And hold forth. Okay, well, this has been a great presentation, David. Thank you so much. I have learned so much, and I am looking forward to seeing your key and using it. I feel like I learn a little bit every year, and you've helped to demystify the blues, at least a little for me. So very much appreciated.
And then you forget it in October, November, December, and then you get to relearn it, and it's
Absolutely,
Oh,
and
yeah.
get all excited. So this is perfect for getting everybody revved up for the new season, because it's going to be within the next few weeks. So
March is the longest Christmas Eve ever.
Okay, well, thank you very much. And next month, we're going to have a presentation by David James about the Western population of monarchs. So feel free to join us the first Wednesday of March. So we'll see you all next time.
Bye.
All right, thanks, everybody. Good night.
Thanks, David. Thank you, David.
Welcome.
Nice job, David. Thank you so much for stepping in. Really, really, really appreciate it.
My pleasure.
Okie doke. See ya.
Good night.
Good night.